Hi, everybody. We're going to be finishing up chapter 10 today. Just share my screen with you. All right. Hello. Okay, cool. So we're done with DNA structure, done with DNA replication. We're going to finish up protein synthesis today, and then after that, do mutations, and then we're done. And next, we'll talk about DNA technology, which is all right. In order to review, go ahead and do the online simulation link on Canvas. It's a little interactive activity. It's a lot of fun, and it doesn't take that long. It requires Flash Player, and I know that some of you might not be able to play it, so I put a video of myself doing it on Canvas, and the quality of the video isn't that great. So if you can do it yourself instead of watching the video, you should do that. But otherwise, you should watch the video. So go do one of those things, and then come back. Today's objective is to be able to transcribe and translate a gene using the genetic code. This guy is the genetic code, this little chart right here. And by the end of today, you will know how to do that. Okay, let's do a quick review of amino acids because proteins are made of amino acids. There's 20 different types, and these are the 20 different types. They all have the same basic structure except for this side group here called R. All of those are different. And proteins are just a bunch of amino acids all strung together and folded up. At this point, go ahead and do questions 1 and 2 of the transcription critical thinking worksheet. Okay, the start codon of every polypeptide is always AUG, which is methionine. Watch the video transcription and translation practice, and as you watch, do the worksheet, transcription and translation practice, and then come back when you're done. This table is called the genetic code, as I have already told you, and it dictates how codons are translated into amino acids. Nearly all organisms use an identical genetic code to convert the mRNA codons transcribed from a gene into the amino acid sequence of a polypeptide. So here you have all the possible combinations of A's, C's, G's, and U's in groups of 3. There are 64 of them. Remember that a codon is a group of 3 mRNA bases, and each codon codes for one amino acid. So using this code, we can decode the information in an mRNA and find out which amino acids are going to be in each polypeptide. Notice that there's multiple codons per amino acid, and because of this, we say that the code is degenerate, or sometimes say we say redundant. So for example, there's 6 different codons for arginine, and this helps reduce the effect of mutations, the impact of mutations. So if there's a mutation in the RNA, it's less likely that it will affect the overall protein, the sequence of amino acids. Check yourself, answer this question, and then finish the transcription critical thinking worksheet. And when you've done that, come on back. All right, COVID-19 connection. So the reason that most people wait to talk about viruses until after protein synthesis is because basically the entire life cycle of a virus is transcription and translation, making its own proteins and exiting the cell. But of course, we didn't do that, so here's a little connection. There's a lot we still don't know about COVID-19, obviously, because it's brand new. But this is what we think the life cycle looks like so far. And a lot of this is based on what we know about other coronaviruses. So in step 1, it attaches to the cell. See, it's got its own surface receptors, and then there's the cell surface receptors. And then its membrane fuses with the membrane of the host cell, and it releases its genetic information. The genetic information in this case is RNA. COVID-19 doesn't have any DNA inside of it. So in order to get DNA, it has to do what's called reverse transcription. Remember, transcription is when the cell reads DNA and gets an RNA copy. But in reverse transcription, the cell is reading RNA and then it gets a DNA copy. And then what happens with that DNA copy is it is inserted, it's integrated into the host cell's genome so that the host cell is copying and transcribing and translating this DNA along with its own. And it doesn't know that it's doing this. It just sees DNA and it's like, okay, I'll make RNA and I'll make proteins. So transcription happens. The DNA is used to make an RNA copy. And then it goes out into the cytoplasm where translation happens. The RNA is used to make proteins. And in this case, these are the viral proteins. And when all the components of the new virus are assembled, they come and they move towards the membrane, and then they bud off and they take part of the host cell membrane with them like that. And this is one of the reasons why hand washing is so effective against this virus, because soap disrupts a membrane. Remember, all membranes are made of a phospholipid bilayer. And you might remember that demonstration I did almost at the very beginning of class, where I mixed oil and water and they wouldn't mix. But then you add soap, and the soap sticks to both of them because it disrupts the separation. Process by which the virus exits the cell is called budding. All right, to summarize, we're done with all of protein synthesis now. Watch the textbook animation Protein Synthesis, which is in the module on Canvas, and then do the concept map that's on the next page. This is from your textbook. I added some stuff. I added this box and this box, this box, and this box. I gave you a little word bank for the ones that have letters in front of them. And the answers are at the end of the PowerPoint for you to check yourself if you want. You should know these things for sure. All right, on to mutations. Mutation is basically a change to the existing DNA. It doesn't have to be a bad thing. There's many different kinds of mutations, and we're going to focus on these four, which are the point mutations, insertions, deletions, and frame shift mutations. There's some overlap between the categories because insertions and deletions especially can cause frame shift mutations. We'll talk about that in just a second. In fact, watch this animation, define these terms as you watch, and you'll probably have to watch it several times because it goes really quickly. But it's an excellent visual of how the mutations will change the DNA structure as a whole. And when you're done with that, come back and then watch Mutations by Mr. Anderson with Bozeman Science. And as you watch the video, answer these questions. I love Mr. Anderson. It is my goal as a teacher to be like him. I do not want to be with him. Some people think that. I just want to be him. He's fantastic. Anyway. Oh yeah, he was voted the best teacher in Montana. Like, Montana's an enormous state. Gosh. Anyway, moving on. Now we're going to read page 203, the fourth paragraph. I'm aware that not all of you have the books, so I've given you a little picture of just this particular page about mutations. Answer, why are insertion and deletion mutations or frame shift mutations potentially so harmful? And the way that you can make this bigger is if you go into PowerPoint. Come on. Over here on the bottom right, you can either zoom in, or you could just take it and like expand it like this. Personally, I find it easier to zoom in, but it is, of course, to you how you want to do it. All right, then watch this animation. It's a TED Ed talk. I love these guys. They're so cool. And answer these questions as you watch. Make sure you know the answers to those questions. All right. All new genetic information, all of it, new alleles, new characteristics, everything, initially starts as a mutation. I don't know what. Okay. It's just a change from something that has existed before. And alleles, remember, they're variations of a gene. Each new allele is the result of a mutation. Mutations are DNA copying errors, and when they happen in cells that produce eggs and sperm, then they can be passed to the offspring as new alleles. When they happen in any other place in your body, they're not going to be passed to offspring, so they won't count as new alleles for the next generation. So, for example, if a mutation happens in one of your skin cells or one of your bone cells, that's not going to be a new allele that's passed to the next generation and gets passed around because sperms and eggs don't come from your skin or your bones. They only come from cells in your ovaries and testes. And a lot of the time people will say, well, blue eyes is a mutation. And yes, it is a mutation, but so is brown eyes and so is green eyes. And which one came first? So which one is normal and then which one is the mutation? Well, it really doesn't matter because they're just variations of each other. And we're going to read this because that relates. It's useful to think of mutation as a process that creates genetic variation. We often refer to mutation as a thing, the genetic variation itself. Its approach can be useful when it comes to a gene associated with disease. However, the disease allele carries a mutation, a DNA change that compromises the protein's function. However, this approach gives mutation a bad name. It's important to remember that losing the function of a gene doesn't always affect health. For example, most mammals have hundreds of genes that code for olfactory receptors, proteins that help us smell. Losing one of these genes probably doesn't make all that much difference. In contrast to variations that cause disease, there are many more examples of variations that are neither good nor bad, but just different, like blood types and eye color. Just like with disease alleles, the process of mutation creates these more neutral versions. But with neutral variations, it can be impossible to tell which allele is the normal one that existed first and which is the mutant, and the distinction is often meaningless. All right, before you take the quiz, make sure you have the answers to all the questions in your notes. And for answers to the concept map, go to the last slide. You're going to need, for this concept map, you're going to need your notes from last time as well. You'll need all your protein synthesis notes. And that's it. Bye. Maybe.